Earth's oceans remain the least explored frontier on our planet. More than 80% of the seafloor has never been mapped, measured, or even seen by human eyes. Tonight, we examine a discovery that challenges what we thought we knew about biology and reveals a life form that should not exist. This investigation begins not with a photograph, but with a signal. On a routine mapping mission, a deep-sea survey team detected something unexpected. A large, stationary mass producing rhythmic fluctuations, breathing patterns. Breathing signatures are normally biological, but this object was hundreds of meters wide. Nothing of that size should breathe. The team assumed a sensor malfunction. They initiated a full system reset. After recalibration, the breathing pattern became even clearer. The anomaly was real. The signal originated from a previously unmapped depression in the Central Pacific. A trench with no recorded biological colonies and no seismic instability. The region was classified as geologically inert, and yet something living appeared to be embedded in the rock. Initial theories included a thermal vent misreading, a pressure-driven sediment shift, a migrating bait ball triggering feedback loops, an unknown mechanical debris signature. None matched the repeating waveform. Something was producing controlled, rhythmic expansion. Something alive. This triggered the launch of ROV-12, a deep submersible designed for extreme depth visual inspection. ROV-12 began its descent at 9:42 UTC. Conditions: stable thermocline, minimal current, visibility low. Its depth sensors crossed 3,000 meters, then 4,000, then 5,200. The anomaly was still more than 300 meters below. What they assumed was drifting sediment was not drifting at all. It was pulsing. At 10:11, UTC ROV-12 captured the first visual confirmation, a faint blue glow emerging at regular intervals from the seafloor. The pulse rate matched the sonar waveform exactly. The team switched to high-gain imaging. The pulses resolved into clear light signatures, following a strict rhythmic pattern. Deep-sea organisms create light, but not like this. Not in coordinated mathematical intervals. Even more unsettling, the light changed when the ROV altered position. It increased in brightness, as if aware of the vehicle. When the ROV closed to 40 meters, the cameras revealed that the glowing mass was enormous, stretching across the seafloor like a living, interwoven mat. Early calculations estimated the organism covered more than 1,200 square meters. The expedition team had never seen a single organism of that magnitude. Vein-like channels ran across its surface. A luminous fluid traveled through them in synchronization with the light pulses. Each pulse appeared to initiate a coordinated contraction, similar to a heartbeat, but spread across an entire biological colony. Still, no known colony organism behaves as one unified structure. This one did. Sensors detected localized pressure fluctuations around the organism, not dangerous, but consistent, as if the structure was modulating the water around itself. This triggered the mission's first caution advisory. The organism was not only alive, it was reactive, and it was aware of the ROV. At 10:22 UTC, the organism released a low-frequency vibration that propagated through the surrounding water. Camera feeds flickered. Seismic sensors spiked. Electrical readings drifted. This was not passive biology. It was active environmental modulation, something no known marine organism performs at this scale. Mission Control halted the descent. The team reviewed telemetry, uncertain whether they had encountered a defensive reaction or an attempt to communicate. No conclusion was reached, but the mission proceeded. Because this organism represented a biological structure never before observed, with cameras stable and interference decreasing, the team prepared for the next step: sample collection. The question was not whether the organism was alive, but what kind of life it represented. ROV-12 initiated a controlled extraction of superficial tissue. The organism did not retract or resist. It simply pulsed, observing. The sample was secured in a sterile containment capsule for ascent. Even separated from its source, the sample continued to pulse, at the same interval recorded on the seafloor. Upon recovery, the sample was transferred to a Level 2 marine biology lab on board the support vessel. Strict isolation procedures were followed. No known pathogen or parasite was suspected, but the organism's behavior warranted caution. The first microscopic examination revealed the structural anomaly that transformed this discovery from unusual to unprecedented. The organism's genetic material was arranged not in a double helix, but a triple helix nucleic structure. This configuration has no known precedent on Earth. This structure would fundamentally alter replication, mutation rate, and molecular function. It suggested not a new species, but a new biological system. Further testing showed rapid regenerative capability. When cells were damaged with micro-incisions, they repaired themselves in seconds, faster than any regeneration process documented in deep-sea organisms. The tissue was subjected to pressures simulating 6,000 meters below sea level. It remained structurally intact. Even more surprising, it demonstrated increased pulse rate under pressure, as if optimized for extreme environments. The most striking property emerged during EM field testing. When exposed to low-frequency electromagnetic waves, the tissue responded immediately, increasing its bioelectric activity. This was measured as accelerated pulsation, increased cellular motion, and synchronized chemical shifts. The organism was not merely alive, it was reactive to electromagnetic stimuli. This finding had no biological explanation. Nothing in Earth's deep ocean requires sensitivity to electromagnetic waves. Light, pressure, heat? Yes, but not radio frequencies. This suggested an evolutionary path completely independent of known biospheres. Genomic sequencing was initiated. Early analysis identified repeating sequences that did not match terrestrial genetic coding. Some patterns resembled self-correcting algorithms, structures that maintained integrity even when simulated mutations were introduced. Such mathematical precision is rare in natural DNA. The implications were staggering. A late afternoon briefing summarized the findings: triple helix nucleic structure, advanced regenerative capacity, high-pressure optimization, electromagnetic responsiveness, synchronized colony behavior. No marine organism, past or present, shared these traits. The working hypothesis became clear. This organism represented a biological domain distinct from all known life on Earth. After sample retrieval, the ROV returned to the anomaly zone to monitor environmental stability. What it found raised even more questions. Schools of fish maintained a consistent 20 to 30 meter distance, as though blocked by an invisible field. This behavior had not been observed in previous deep ocean surveys. Crabs and eels that approached too closely froze in place, as if momentarily stunned, then retreated with rapid, erratic motion. Within hours, scientific instruments placed near the organism began to fail. Power cells drained faster than expected. Compass readings drifted several degrees. Magnetic field sensors fluctuated unpredictably. These disturbances were localized, confined to the organism's immediate vicinity. A high-resolution magnetometer grid revealed mild but consistent magnetic distortion. It pulsed in synchrony with the organism's light emissions. This was not random environmental noise, it was a signature, a repeating pattern linked directly to biological behavior. Temperature probes recorded subtle, rhythmic decreases in water temperature, just a few tenths of a degree, but synchronized with the organism's pulsation. No known organism regulates its environment at this scale. Sequential mapping showed the organism expanding by measurable amounts, between 4 and 7 centimeters per week. This rate exceeded projections for deep-sea microbial mats by an order of magnitude. The organism was not static. It was spreading. The deeper the team studied the region, the clearer it became. The organism was influencing its ecosystem and possibly defending territory. Researchers debated possible explanations: chemical emissions, low-frequency resonance, electromagnetic interference, thermal microgradients. None fully matched the observed patterns. The organism was altering its environment in ways not previously recorded in marine biology. This led to the mission's most difficult question: was this adaptation or communication? Following the initial findings, a multi-institution briefing was held involving marine biologists, molecular geneticists, geophysicists, and astrobiology experts. Their goal: to classify the organism and determine where it belonged in the tree of life. The conclusion was unanimous. It did not belong anywhere on that tree. The team began developing theoretical models to explain the organism's existence. 3 primary hypotheses emerged. The first theory proposed that the organism was a survivor from an ancient, isolated ecosystem. a lineage predating modern multicellular life. Earth's deep oceans have remained stable for millions of years, providing refuge for life forms that evolved under extreme pressure and isolation. But this theory encountered a significant flaw. No known ancient organism possesses triple helix nucleic acids or electromagnetic responsiveness. The discrepancy was too large to ignore. A second theory suggested the organism represented a third domain of life, separate from eukaryotes, prokaryotes, archaea. This would imply a parallel biological framework with its own genetic coding principles. If true, Earth may host more than one origin point for life. The implications would rewrite fundamental biology. New genetic architectures, new biochemical pathways, new evolutionary pressures. This was the theory that most intrigued the geneticists. This finding complicated the second theory. Furthermore, field observations pointed to behavior inconsistent with isolated evolutionary development, which led to the most controversial proposal. The third theory postulated that the organism did not originate on Earth. Several possibilities were considered. Biological remnants delivered by meteorite, survivors from panspermia events, organisms adapted to extreme environments similar to oceanic trenches, a transferred fragment of an unknown biosphere. The structure of the organism's genetic code and its electromagnetic sensitivity supported the idea of evolution under conditions not found on Earth. Some researchers believe the organism's pulsed light patterns and electromagnetic emissions might not be physiological processes, but communication methods. These pulses occurred too regularly, too precisely, to be random. But communication with whom, and for what purpose? No answer has been found. With growth rates between 4 and 7 centimeters per week, the organism showed potential to expand across entire trench systems. Models suggested that in 20 to 30 years it could cover more than 10 square kilometers of seafloor. Environmental impact projections included disruption of marine migration patterns, ecosystem displacement, alteration of local magnetic fields, unknown long-term chemical interactions. A full risk assessment was initiated. By this stage, the research team understood one thing clearly. They were studying something beyond known Earth biology. Despite extensive analysis, fossil history provided no clues, genetic data offered no terrestrial match, and ecological behavior broke all known biological rules. The organism's origin remained unresolved. Whether it evolved on Earth or arrived from somewhere else, researchers could not determine. Let's summarize what has been confirmed. 1. It is a single organism covering over 1,200 square meters. 2. It displays coordinated colony-wide pulsed behavior. 3rd, its genetic material exists in a triple helix configuration. 4th, it reacts to electromagnetic fields instantly. 5th, it alters its surrounding environment. 6th, it shows steady, measurable expansion. These observations alone challenge modern biology, but combined, they suggest something unprecedented. Even after weeks of study, the team could not categorize the organism, not as bacteria, not as fungus, not as animal, not as plant, not as any known domain of life. It remained unclassified. The deep ocean covers more than half the Earth's surface and remains largely unexplored. If an organism of this scale, this complexity, this strangeness can exist undetected for millions of years, then we must ask, what else is hidden in the abyss? What other life forms operate beyond our understanding? And how many discoveries waiting in the dark will force us to redefine life itself? We may never know where this organism came from, or what its presence means for Earth's ecosystems, but one thing is certain: we are not finished discovering what lies beneath us. Thank you for watching Dark Science Files. If you want more long-form investigations, subscribe, because the mysteries only grow deeper from here.